right, welcome back. We're going to continue on with Chapter 18 stuff, talking about how organisms are classified and grouped together and how we create hypotheses using cladograms. So, comparing form and function for Section 3, physical similarities are often evidence of shared ancestry, but sometimes a trait evolves independently into groups, which we'll give examples here later. In many cases, comparative morphology can be used to unravel evolutionary relationships. Again, morphology is great for classifying organisms and seeing how closely related they are, but as we talked about before, you cannot solely use morphology. Sometimes these traits can evolve independently and as a result give you misleading hypotheses. So, morphological divergence. You have homologous structures, and the word homo in science means the same. So, we're talking about same type of structures in an organism. Structures that are similar body parts in separated lineages that evolved in a common ancestor. So, the ancestor evolved these traits and they were favorable. They were used and they were passed on to its offspring and all the descendants afterwards. So this wound up in many organisms later on down the road. Now, they may have changed a bit, but they are very similar. So homologous structures may be used for different purposes now, but the same genes direct their development. So the genes for these particular things still exist. Morphological divergence, change from the body form of a common ancestor, is an evolutionary pattern. For example, vertebrate forelimbs vary in size. Vertebrate forelimbs vary in size, shape, and function, but are alike in structure. So as we look at this, so we're talking about the morphology of the forelimbs in things that have vertebrae. So as we look here, we have a chicken, penguin, a porpoise, bat, human, elephant, and whatnot. So as we look at these organisms, we see that, yeah, these forelimbs are totally different. There's no way that they're similar. But as we look at the bone structure, the actual bone structure, again, some of the sizes vary, forms may vary, but the overall concept is still there for muscle attachment and movement a lot of times. As we look at the human arm, something that we're familiar with, we have the humerus here, which is from your shoulder to your elbow, and then you have the ulna and radius, which are the two bones that allow for that rotation in your forearm. Then you have your carpals, your metacarpals, and your phalanges. So, pretty simple. Now, we look at the reptiles, something that we believe that reptiles gave a lot of these traits and stuff to us because they were previous ancestor to many of these things. As we look at the reptile, we see too that it has a type of humerus, an ulna, a radius, carpals, metacarpals, and phalanges all within its forelimb. Now, that's a much older ancestor, but as we look at a bat, something that flies through the air compared to us that uses our arms to grab and move things around, they're used nearest to fly, but they too have a humerus, an ulna radius. Now, if you look, this bone is much thinner than say ours would be, but it still exists. Okay, still has carpals, metacarpals, and its phalanges are much longer, making those wings or that skin between the fingers essentially to allow for flight. So different purposes for what they're used for, but they still exist. You know, we look at a penguin, for example. They too have this. A chicken as well, and even an elephant, even some large animal that uses all four of its legs to walk around, still have all these bones within them. So these are homologous structures, indicating that perhaps they came from a common ancestor long, long ago, and these favorable things used for walking on land were, well, again, genes that were passed on to several different organisms, as you can see here. So scientists wanted to figure out how closely related are these organisms based upon these morphological traits. Another interesting thing is in embryo development. As we look at the developing stages of an embryo inside the womb there, we notice that there are several common trends, that there are genes that help develop that embryo into a living, viable offspring one day. And some of these genes have been conserved because if they change them too much and or it just doesn't work, the offspring is never made. So these genes tend to stay highly conserved. Now, one interesting thing that we find among all sorts of developing embryos, fish, reptile, birds, and humans, is that they all have these gill slits, not gills, slits for gills. Okay, so a totally different thing. But again, you know, fish, they're going to need gills. But a reptile, no, they don't need gill slits. Nor does a bird or a human. But yet we all have them in our pre-stages of embryonic development. Now, eventually, in the reptiles, birds, and humans, these gill slits disappear due to the process of apoptosis. They're no longer needed because, again, we don't need this. We're on land. So again, some of these genes are conserved and are there, but not necessarily used in adulthood or even infancy stages. But again, there's things like a tail in the fish. Reptiles, some of them have tails. Birds, they have little nubs, but yeah. And even humans, in our embryonic development, we have tails as well. Again, apoptosis deletes that out, but there are some humans born with tails each year. All right, so these genes are there, and they have been there. So we use genetics as well to help develop our understanding of how species evolved over time. You got morphological convergence. You have analogous structures, body parts that look alike, but yet did not evolve in a shared ancestor. They evolved independently in lineages subject to the same environmental pressures for whatever reason. You also have morphological convergence, the independent evolution of similar body parts in different lineages. For example, you have birds and bats and insect wings. They all develop the ability to fly. However, that ability to fly was an independent evolutionary trend. All right? It was not that they all got the ability to fly from one direct common ancestor. They may have a direct common ancestor way back in the line, but doesn't mean that ancestor flew. They could have walked on all four and then given rise to these individuals later. Okay, so totally different. So as we look at the picture here, we can see that insects, bats, and birds are all flying. As we look at our cladogram down here, we can see that birds and crocodiles, they form a more closely related clade or sister taxa up here. Birds develop the ability to have wings later on, separate from other organisms within their sister clade. But yet we see that birds, humans, crocodiles, and birds all have 5 digits. The insects do not. They still evolved the ability to fly independently, and so did birds. So again, we're looking at the most parsimonious analysis that we can for all of these traits combined together. So morphological convergence, we see some of these interesting little things happen. As we look at this picture up here in the left-hand corner of the butterflies, we see that lovely monarch butterfly. However, the one on the left is a viceroy butterfly. It mimics what the monarch looks like. So the monarch butterflies, they have a very distasteful taste to them when a predator eats them. It can make them sick, throw up, not feel good, and stuff like that. So many birds and other animals try to stay away from these or eating these. However, the viceroy doesn't have that undistasteful taste to other organisms. However, it looks like the monarch butterfly. So just looking like something can actually deter predators from eating you. Now, did the viceroy butterfly choose to have that orange and black? No, not necessarily. There was probably a variety of butterflies out there, and they happened to have a trait for orange color. Being they had that orange color, birds were like, I don't know, maybe I could eat him, maybe I not, I don't know. So they kind of stayed away. Well, some of them took the risk and they ate them. But however, some of these butterflies had these unique lines on them and their wings to resemble more the monarch butterfly. And over time, they were naturally selected for. And today, you can actually see there are subtle differences between the two. One big difference is this band on the lower wing here. There's a line. That's how you can tell a viceroy from a monarch. Over here, you can see the eastern coral snake and the scarlet king snake. The eastern coral snake is a very, very venomous snake. You do not ever want to pick this thing up. So be careful. But it has that black, red, and yellow color. However, the scarlet king snake is one of the most docile, non-venomous snakes you'll ever meet. They're very, very pretty. Absolutely stunning if you ever see one in the wild. But they do have those bright colors, the black and the yellow and the red. But again, they're not venomous. They just happen to look like this, and a lot of animals tend to leave it alone because, bright colors in nature tend to stand out and say, "Whoa, hey, don't mess with me," and they just happen to be left alone because of it. And that's their natural selection there. The frogs down here at the lower right, again, bright colors indicate poisonous or dangerous, but yet, you look at these, you're like, yeah, they look really similar, but they could be totally different frogs, which result in just a commonality between colors. One of them is venomous, one of them is not. Over on the left, you got the wasp, which is, as we know, very frightening, that they can sting you. But then you got the clear-winged moth over here that happens to be yellow and black and have clear wings. Would never hurt you. It couldn't if it tried. So, but yet it tends to look like it. So birds and other animals tend to leave it alone as well. So convergence. So take-home message: what does comparative morphology reveal about phylogeny? In morphology, divergence of a body part inherited from a common ancestor becomes modified differently in different lines of descendants, homologous structures. In morphological convergence, body parts that appear alike evolved independently in different lineages, not in common ancestor, analogous structures. So the next part what we're going to do is talking about the biochemistry and also some genetics. So we're going to stop here today, and I'm going to show you your assignment. So you can open up a Google tab here and type in the word GenBank, GenBank, and it should take you to the first website here, right here. And this website has a data bank or storage of all sorts of DNA sequenced genes, genomes, whatever it may be, from all sorts of scientists across the world. So they can submit their work and their codes that they sequenced to this website for all scientists to use. So it's just one giant data bank. You can find entire genomes for humans and other things like that out there. No, they're not going to have names to them and whatnot, but yeah, they have some really cool stuff out here. So what I'm going to do is I'm going to type in a very similar thing that I think I talked about before. Hortus, which is the scientific name for a rattlesnake, so Crotalus hortus. Um, this website likes to deal with more scientific names rather than just common names like the red fox or something like that. So try to use the scientific name you can. And what I'm going to do is I'm going to click on it. It says Crotalus hortus, which is the rattlesnake. So I'm going to click on that, and it tells you there's 399 base pairs linear DNA. So as I click on this, it will open up another page. It will give me some information of who uploaded this, the journal article in which they wrote about it, and you can see the actual DNA code right there, and they've got it in lines. So that's pretty cool. Now, what is even cooler is you can actually compare this DNA fragment for this particular gene or whatever to many other ones out there by simply running a blast. So on the right side here, you click on Run Blast. It brings you to another page here. Most of this you just leave alone. But what you can do is if you want to look for, say, similar sequences for other rattlesnakes, we want to see, well, other rattlesnakes out there. You can do highly similar, which means the exact same species pretty much, or you can do dissimilar, which you're going to get a little few different species out there, maybe some that are closely related but not necessarily the same genus. Or you can do somewhat similar, so whatnot. I'm going to choose somewhat similar just because I want to look at the rattlesnakes. And I'm going to go down here and run blast. When this does this, it'll take a few seconds to run depending on how many results they find and how big the gene is. If your internet's really slow, it may take a lot longer, so just give it a few seconds and it should work. Unless, like I said, you have a really big gene. Try to choose a gene that's probably like under 500 base pairs if you can for your assignment today. And it'll pop up with this page. And as you scroll down, there's got a lot of technical jargon up there. But it's gone through the GenBank website and it blasted out a search for similar genes out there. And it found all of these results here. And there's about 100 of them. And you can see there are scientific names. Some of these genes are similar. You may get to some, and later we'll talk about, that are predicted. It's basically looking at, like, say, genomes of certain organisms, and they find embedded genes that are similar. They think that it's it, but they're not sure. So you can use them, but you don't have to. So as we look at this, I'm going to go ahead and deselect because it's got all of them selected. And I'm just going to pick a few of them, and I can see there's Crotalus hortus. I'm going to try to find some different ones like Viridis, this one, and this one. And just get a few of them here. So now I've got a couple here, and what I'm going to do is I want to run a distance tree as a result. So they're trying to create a parsimonious analysis for the ones that you selected, and they're going to build a cladogram. So as I click on this, it should open another tab for me. And it should quickly take those individuals that I had and run a result. So it shows me the one that I chose to begin with, and you can see it's built this cladogram, and it's got little nodes on it showing you where the common ancestor is and how each one of these is closely related based upon that gene that you selected, not necessarily as a species, but the gene that you selected. So as we look at this, we can see that the Crotalus hortus, it matches up with the Crotalus hortus. Then we've got these other ones that are a little different. So they're different species, which is kind of cool. And you can see how each species is somewhat closely related or dissimilar based upon all this information. Now the longer lines and stuff means, passive divergence. So if they're not much of a line, that means they're, pretty recent, pretty close. So, but that is pretty cool. So that's your assignment, is to take a blast and for a species, a gene, whatever you want to do, and get a screenshot of this. So if you're using your computer, you can use the snippet tool. If you're using a Chromebook, you can always do that control window change, and it should take you a little snippet or a screenshot of this. You can always take a picture with your phone, whatever, and upload it into the assignment page. Okay. Now, if you want to do, say, a gene, because you can look at just genes instead. So I chose to look at the Homo sapiens BRCA1 gene, which is this is the breast cancer gene. So if you're looking into something like that, you can always look at that. So this particular one, it's ran a search for me, and it's got some ones up here that are not necessarily humans, but if I scroll down, I'll find some. And here's the one I clicked on before. And this one has the gene within it, and it's not super long, it's pretty small, so just a small segment of it, whatever it may be. So it may not be the whole gene, just a little bit. And I'm going to run a blast for this. So as I do this, I want to see how the breast cancer gene is correlated to, say, other organisms, not necessarily humans. So I'm going to click on, this time, more dissimilar sequences. So maybe I can get a wide variety of organisms, not just, say, Homo sapiens. Run the blast, and it should take a few seconds again. Shouldn't take too long. If it does, just go get you a drink of water, go for a walk, come back, and it should pop up. But scientists have been using this for a long time, and each year it tends to get better and better with the tools that they can use, the search. And as more scientists put more and more data out there, it just becomes a pool of information that we can draw from. So there are countless of entries out there that can be studied all the time. So you could almost write your own scientific article using other people's work in DNA. You don't even have to go out and find it. You can just find hidden things in there. It's kind of cool. So now we've got the Homo sapiens, and I'm going to go ahead and select one that's very similar to the one we selected, just so we can find it. And then we got Homo sapiens, and then we've got a whole bunch of Homo sapiens. So we still got a lot of human species out there, but then all of a sudden we get down to Pan-Troglodytes, which is a very similar base pair recognition here. We can see the percentage breakdown, but here's the crazy thing. This is not a human. This is another primate. So I'm going to go ahead and click on it. And it does say predicted. Again, that's because they're looking at maybe a bigger subset of DNA. So they're trying to say that this might possibly be the gene you're looking for. We've got another one, Panpeniscus. We got some other ones, Homo sapiens. Let's see if we can find a few more. Got another Pan-Troglodytes, we'll get to him. And then we've got the gorillas down here, predicted again. And then we'll go ahead and get this one down here, and we'll grab another gorilla too, just for fun. Okay, so now we're going to build our tree to see how all of these are connected to this gene, see the evolution of this gene, or how similar they are per species. So as we look at this, there we go. Not much in the difference here. We've got gorilla, which that should match up, same species, so they should be pretty similar for that species. But then we got this one here, Panpeniscus, Homo sapiens, Pan-Troglodytes, and Homo sapiens again. So they're all very similar, except for this one right here is a little bit different. So there's a little more divergence within this cluster here, the sister clade. This is the Pongo. So as you can see, there's some diversity among the gene itself. Now that doesn't mean that's how the species evolved per se, or how closely related they are. It's just how that particular gene is affecting them. Okay, so there is two examples, one for species, one for just a particular gene. But your assignment again is to figure out a species and or gene and run the blast, and then select a few of them, probably no more than 10, probably less than 10 would be best, and build that distance tree so you can get this, and then take a screenshot and upload it. Okay, I'm sure there'll be some questions, so I'll be available email. So just email me any questions that you might have. And again, there's no immediate due date on this, so take your time, and don't worry or stress about it too much. Okay, thanks for tuning in, guys. I really appreciate you guys listening to all this stuff and furthering your education.